I will just give you an overview of our tomographic methods. This includes also some of the technologies that we are using in PhenoRob project or projects that are related to the PhenoRob project. Okay, so as you may know that in Jülich we have different facilities for screening plants, starting from the seed to the seed establishment phase and the vegetative growth rate, and also for yield and harvesting products. The technologies that are shown here are more high throughput methods. For example, maybe you're aware of the Rhizo3 project, Rhizotrons here, where we can screen lots of different, up to 900 shoots and parts of the roots in Rhizotrons. But today I would like to focus on our tomographic methods. So we are using CT, for example, for seed phenotyping, in-depth seed phenotyping. And the other tool that we are using is magnetic resonance imaging, MRI, and positron emission tomography for investigating the carbon fluxes in roots. And I would like to start with an example of our seed phenotyping facility. We have different options here to scan and screen seeds, and our motivation was to start the seed phenotyping approaches was to reduce the statistical variance of our starting material, right? So if we have these high throughput phenotyping activities, it would be good to select and be aware of different seed traits for example, making some co-analysis here, or select the specific seeds that are suitable for the phenotyping approaches. And also at the end of our phenotyping pipeline, we were also interested in characterizing the harvest product, like from the start of the plant growth up to the end at the harvest, characterize the seeds. And on the other hand, we were also interested, or we are interested, of course, in the physiological part of the seeds, and to identify key seeds properties, and then also to select seeds for further treatments or for the next generations. And as you can see here, as one of our facilities, the so-called PhenoCida, which is a pick-and-place robot system where we can screen different seed sizes, starting from the small seeds, the Arabidopsis, to rapeseed, barley, and wheat, or also corn seeds. And the traits that we have established at the moment here are, for example, projected areas, volume, mass, density, and color information, or 3D shapes. And this is all done on an individual seed level. And after the phenotyping the seeds, we have also assessed different options to treat those different seeds. For example, we can collect different seeds as numbers in batches, or we can sort them by color and weight. Or, for example, we can just store them individual for further treatments, or just directly plant them on soils. And more recently developed assay is that we can first store them and then do some quick planting. And as you can see, those traits that I just presented here are all biometric traits, so from the outside. And now what we are trying to do is doing the next level of phenotyping. So we also try to look deep inside of those seeds. So with the seed CT, we are able to look deep inside of individual seeds. This is a rapeseed here, for example, to separate the size of embryos or cotyledones. Then we can increase the throughput and screening some more seeds at the same time. Or also, and this is our current approach, to scan different batches of seeds. This is actually Arabidopsis seeds, so really small seeds where our system is at its limit, and then compare different batches of seeds and then go to more individual analysis of seeds within these kind of populations or within batch distribution. Since it's a CT, it's also open for other examples or other objects that are we interested in. And one aspect is, for example, we can also look inside little twigs like this one here, and then we could display the cavitated vessels of this twig. So we can really look inside, non-invasively, actually inside, and determine, for example, the vessels inside of a twig. And another important information, of course, and this will be also my link to the root system, are that we can also look inside of roots. And this is an example of a root that was excavated, and you can see here some cuttings from different roots, primary root and some lateral roots, and we can then screen through the root system and see, for example, how the roots are merging with each other and have some detail look inside of the root systems. In general, we are using not CT for root imaging, and we are using MRI for roots screening. And this brings me to the next larger part of my presentation, and this is the below ground part of roots. So we are all familiar with the above ground part of the plant, with the shoots, because we can easily clamp our machines on the shoot part, and also with remote sensing, as we're doing also in PhenoRob. It's relatively easy to access, although there are quite a bit of challenges there. But then to look also below ground is even more challenging, right? But if you can see the structural information, or the structural diversity, and potentially also the functional diversity below ground, might be also as high as the above ground part of the plants. But it's so difficult to access, right? So the easiest way would be just to simply turn around the root system and have a look and clamp our machines on this, but it's not possible. And therefore, we are using two different technologies that are non-invasively to follow growth of individual plants. This is on the one hand magnetic resonance imaging, and on the other hand, for carbon allocation and how this is fueled into the root system, we are using positron emission tomography. And this is what I would like to show you some examples in the next slides. Currently, we developed some higher throughput systems. So we have our magnet here, and at the moment, our automation allows us for screening 70 plants. So we have a robot system that automatically transfers the plants from a climate chamber into the magnetic resonance imaging device, the magnet, right? And what you can see here is how the robot system transfers the plant into the magnet. And this is some of the images that we could obtain from the MRI. This is for shoots and for roots. And you can see with a high frequency of measurements, the development of plants in such a pot. The current standard pots are 40 centimeter length and 9 or 12 centimeter width. So this allows us to scan roots and shoots of a plant for about 3 to 4 weeks. And one example how we can use this was a nice paper together with Gwendolyn Kirchner from the Frank Hochholdinger lab, where Frank and Gwendolyn identified some genetic genes that were important in the setup of root angles, for example. And we could show that also in natural field soil, these kind of mutation appeared, and we could really nicely show differences in between the wild type and the mutants. And as you know, that this root angle is important for capturing nutrients from soils. We are quite happy that we could also show this in soil in natural conditions. Another example is, and it's really important for us, it's not only to show the difference in images and in videos, but also, of course, to quantify those differences. And as you can see here, an example for the growth rates, we can see a high spatially and temporal differences of how a root grows, where newly formed roots are emergent, and also how within the root system, how heterogenic these kind of growth patterns are. Another example is that we can show diameter classes The color code gives you different areas of the root systems and different diameters, or also age dependency of root systems. So we can see also here as strong differences, spatial temporal differences of the root systems. But imaging is also one thing, but of course we can just get the data and then do our analysis in different tools like R or Python or whatever you would like to do. Another issue or another challenge is always that we are asked, what kind of soils are you using, right? So what kind of, is it a natural substrate or is it just very artificial substrate? And to answer these kind of questions, we ordered from the LUFA in Speyer, so the Landwirtschaftliche Untersuchungs- und Forschungsanstalt, and they provide commercially available standard soils. And we compared those soils with the standard soils that we were using for MRI measurements, right? And then we imaged with our standard protocol the roots growing in these kind of soils. And we found that out of these 6 available soil types, different soil types, at least 4 of those were really well suitable, or 3 were really well suitable and had the same quality of data analysis as our standard soil that we're regularly using in the MRI. 2 others were also nicely visible and analyzes, but with another cutoff, so we could not detect really small root sizes here, root types, and only one soil, which was really cleanly loamy soil, we were only able to visualize the primary roots, for example, where we would say, okay, in this case, we would not use these kind of soils. And from our experience is always that we know we have some access projects also to use the MRI. The best thing from my point of view is always, if you want to collaborate with us, it's always the idea, let's test the soil first and make some preliminary pilot studies and check whether the soil type is suitable for the MRI. Another challenge is the throughput, right? So I just told you that we have now a kind of an automated system for about 70 plants, but we can even go higher. My colleague Daniel Flutfelder, he set up an experiment where he was interested in early vigor and early emergence of plants, and he established these kind of towers here, where each of those towers, or let's say levels, could be run with 18 seeds. And he was able to segment those seeds here and identify on a high temporal level frequency to scan the roots inside of these kind of towers here. And we were able to scan in this experiment up to almost 300 seeds, seedlings, about several days. And what he found out after segmentation of the seeds in these kind of pots, that already at early time points, the different genotypes behave quite different. So we have here in solid genotype 1 and in dashed lines the genotype 2. This is a subset, and you can see that, for example, for root lengths, we have already at these early time points, so starting at 6 or up to 66 hours after emergence of the seed, quite some differences in the behavior, in the growth behavior of those different genotypes. Also for growth rates, for example, you can directly see here in red, for example, for the primary root, that it's not linear, but quite different and quite heterogeneously how these roots are growing and the plants are emerging in soils. So with this was, for me personally, a really good example how MRI can be used for a high frequency of measurements and early development of plants in soils. These examples I'm showing you gives you a good overview over the structural information that we can obtain using MRI. On the other hand, of course, we are quite interested in the functioning of plants, right? For example, what our aim is here to visualize and quantify assimilates allocation. So recently fixed photosynthetes and how they are distributed in the plant shoots. And here in this presentation, I will focus on the plant roots because about 60% of the photosynthetically fixed carbon is allocated to roots. And there it's used to build up biomass and the metabolism and growth of the roots. But on the other hand, part of this carbon is also excreted into the rhizosphere, where it fuels microbial communities, the activity of those and the growth. And in the past, and I think you are aware of this, these kind of tracer studies has often been done with 14C and 13C. We are using a different isotope at the Forschungszentrum. As the Forschungszentrum is a former nuclear research center, we can use 11C. And 11C is a PET isotope, so we can use it in positron emission tomographs to visualize carbon allocation in a 3D environment without destructive harvests. The important thing to understand here is that this kind of tracer has a half-life of 20 minutes. And this means that after several half-lives, the radioactivity is gone, and we can then use these plants again for further analysis, and it's not invasive. So it allows us for repeated measurements of individual plants and determination of carbon tracer allocation and transport velocities within the root system. And this is what I want to show you in the next slides. First of all, I mean, the repeated measurements and are based on the short half-life. But the half-life of 20 minutes has also some challenges. And this means that we need to produce this tracer on-site. So we have not much time to lose in order to start the measurements. And therefore, we are running our own cyclotron for producing the 11CO2. We also have a sophisticated plant exchange measurement system and also our PET tomograph here, the PhenoPET, where we can scan the root system and also the shoot system in 3D. And this is how it looks like. We have here as a pea plant as an example, where we can label the shoot with CO2. And then the plant takes it up and transfers it from the shoots to the root system. And how a typical image of a tracer looks like is this. This is a pea plant, and you can see how now the tracer fuels into the root system of the plant. And this is a pea plant, so it interacts with some rhizobia and forms the nodules. And these dots here, these are the nodules of the plant. This is the place where the rhizobia fixes the nitrogen from the air and in turn receives recently fixed carbon from the plant. So these dots here are displacing hotspots of carbon assimilation and also displace strong carbon sinks. And my colleague Ralf Metzner, he managed to identify in our analysis pipeline, he identifies this region of interest here within the 3D environment of those nodules. He collected those information of the specific activity of the tracer inside the nodules, and then he asked the question, what happens or what will happen if we add now mineral nitrogen to the root system? Meaning that the plant is able to take up this resource via its own root system and is not depending anymore on the rhizobia symbiont. So would the plant reallocate their carbon resources? And what would happen after this? And what you can see here is the tracer fraction before and after the addition of the nitrogen resource. And you can clearly see that the fraction of carbon allocated to the nodules was reduced after the addition, but it was increased to the root system. So the plant was at least partly able to reallocate the carbon allocation according to its needs on a short time interval. So really affect this nitrogen addition affected the reallocation of carbon on a short time scale. And now we have the opportunity, we have 3D information from the structural part of the plants, from the plant roots, and also from the functional plant, and we can combine those images, right? We can do a 3D co-registration. And this comes now to a part which is an example that we are using also in PhenoRob, in the Open Call 2 project. This is a PhD thesis of Cabena, and in cooperation with Ann-Kathrin and Justus Detring and Christoph Braukagel and Pira Pira. So we are interested in the carbon allocation within a sugar beet. And as you can see here, we labeled a single leaf, and these are 3 independent labeling, and you can see that the carbon allocation is not homogeneously within the beet of the sugar beet, but what we can see is that there come sectors of how the plant allocates the recently fixed carbon resources into the sugar beet. And the challenge now is that with Ann-Kathrin and Justus, we are trying to understand how the plant responds also on the shoot level to a biotic stressor, and we are investing now how this allocation pattern changed according to a biotic stressor, which is affecting the phloem transport in the sugar beet. And we just this morning received the message from Justus that the inoculation with the SBR, this is a pathogen we are using, was quite successful, and we hope that in the near future we can send you or can show you some more information on how the stressor affects the carbon allocation below ground, also how it's affecting the shoot part of the sugar beet. So we are trying to combine the shoot and the root information of the response to a biotic stressor. And to answer also this question from the below ground, of course, we need to adapt our system so we can now easily and non-invasively get some information on the ring formation, on the structural information, and also identify some internal structures that hopefully gives us some more insight in the mechanistic responses of plants to these kind of stresses. Another example is how we can use those combination of MRI and PET is when we look into different phenological steps of a plant. With Claudia Knief and Sina Schultes in another project, we were investigating how the carbon allocation is changed in time in maize plants. So in gray, what you can see here is the structural information from the maize roots at 6, 13, and 20 days after emergence. And in pink, you can see the carbon tracer within the root system. And directly what you can see is there's a strong heterogeneity in time and also within the root system. So in the beginning of our labeling procedure and our imaging, the phenotyping, we were quite surprised that now carbon is viewed below ground. First, we saw that something happened with our PET system, but we could nicely see that there is kind of a PET signal here on the shoot part, but it's not transferred below ground. And most probably one of explanation is that at this time point, at early time points, it's quite clear that the seed is still fueling the growth of the root system with carbon reserves. And the plant is focusing in establishing the above ground part of the plant. And then in the next, you can see that this kind of carbon allocation differs within the root system quite a bit. So there is a shift in allocation below ground. And Sina, she managed to link these allocation patterns to different root types, right? She could now identify at the different time points, the time series, how fast and with what kind of intensity the tracer was allocated below ground. And she's now using this kind of information for understanding the carbon allocation and using this for image-guided sampling. So she now wants to identify the microbial community according to the tracer level and how much this tracer level explains the microbial community composition. So the relevance, for example, here is an example for the microbial community where she just said, okay, we have the photosynthase level for low, medium, and high, and how this structures the microbial community. It's just a kind of a kind of an impression, and in the near future, a lot of more of this kind of information is coming up from Zena. And on the other hand, as I said, we can also determine the transport velocity of the tracer, how fast the recently carbohydrates are transported in the different root types below ground are performed. And you can see here on the color code in blue the primary root, and in orange the seminar roots, and in pale or white you can see the nodal roots of the first generation. You can see that the transport velocities within the different root types differs quite a bit. And also in time, these kind of velocities are increasing. Yeah, and with this, actually I was quite fast with my presentation, I think. I'm almost at the end already. So I would like to conclude with that I hope I could give you already a nice overview how we're using these kind of tomographic methods in IBD2 and also in other projects like in PhenoRob, where we're using CT, MRI, and PET for 3D phenotyping of plants. And of course, if you take the temporal aspect into account, we can say that it's more 4D phenotyping of plants. How the structural responses differs in time, for example, root growth, but also how it strongly affects the physiological processes like carbon transport and carbon partitioning in response to abiotic factors and also to microbial interactions. And this gives us a lot of information on plastic response of individual plants, how they respond on a short time span within a root system, for example. And we hope that this gives us the opportunity for alternative trait discovery and then also faster genetic gains by phenotyping to translate this into yield. And my final remark is I have to thank a lot of people, especially I would emphasize, of course, the groups, the enabling technology groups, which is, as you can see, not only consistent of biologists. Actually, the biologists like I am are the minor part of this group. We have a lot of physics, people from physical department. We have computer scientists from the engineering part in here. So it's a true interdisciplinary group to run these kind of facilities. I think it's also necessary and of high value. And I would also, of course, thank a lot of people from the IBD2 and also the collaboration partners running and giving some insights and open up for questions using these kind of phenotyping facilities. Right? And this is also a message that we are open for collaboration in this. We have in PhenoRob, we have a lot of opportunities. And as you can see, with the example with Ann-Kathrin and Justus, for example. But we are also open for other opportunities here. Yeah, and then I would like to thank you for your attention, and I'm open for questions.